I'm Giovanna Prout. I'm the Director of Strategic Marketing at 10X. And this is really an incredibly exciting time for 10X as we see our solutions be used for so many discoveries in human genetics. So thank you very much for being here today, and we really appreciate such a large crowd. It's pretty awesome to see. So I'm going to kick us off this afternoon. I'm going to give you an overview of the 10X portfolio. And I'm also going to tell you about some really exciting product developments that we just announced yesterday. Then I'll be introducing our two speakers, Molly Gasparini and Dr. Jimmy Yee, who will be highlighting some of their work in single-cell genomics. So let's get started. As you know, biology and human biology is immensely complex. From the trillions of cells and types in the human body to the genes and the nucleotides that are found in the DNA of those cells, to the structure of the chromatin, to the translation of those genes, which differ per cell type. And further, the complexity continues as we see in systems of the body like the immune system, where cells have a highly adaptive component in response to antigens. The complexity is enormous, yet due to a lot of your work, there are many knowns, but still so much left to discover. An example of this defining of complexity has been taken on by the Human Cell Atlas Global Consortium. Much like the Human Genome Project fundamentally changed the way we explored biology more than a decade ago, the Human Cell Atlas and other initiatives around single-cell genomics, transcriptomics, and epigenomics will again change the way we explore biology, ultimately to influence the treatment of disease. It's 10X's mission to accelerate these studies and reveal these complexities through the solutions that we offer to you. The core technology that drives the scalability and ease of our products and which allows for new and exciting products to happen is the microfluidic system for massive throughput of molecular barcoding and partitioning of reagents into nanoliter-sized reaction vessels. All of our solutions and our new technology really rely on this core technology. Before our announcements yesterday of all of our new products, this is what the 10X portfolio looked like. And it consisted of our first implementation of this technology that I just showed you. We released our linked reads for genome and exome and de novo assembly, and then extended this technology for single-cell capabilities with single-cell gene expression and single-cell immune profiling solutions. And most recently this year, innovating on that core technology, we released our single-cell DNA solution with a CNV application. And although methods like genome and exome sequencing, RNA-seq, and whole genome CNV analysis have been well-established NGS methods over the years, 10X Genomics has taken those solutions and improved on them and made them accessible to use for anyone for truer resolution and dimensionality. I'll tell you more about our existing portfolio before getting into some of our new and exciting products. For genome, exome, and de novo assembly, the linked read technology enables long-range genomic information from ubiquitous and reliable short NGS reads. It reveals the complexity of genomics, like complex structural rearrangements or deciphering from which haplotype disease causal variants come from, which are really critical in the study of disease without the additional investment in other NGS systems. Now, in addition to high quality and resolution genotype information, we also need high resolution phenotype information in single cells. Gene expression analysis is a standard and robust tool for understanding the transcriptome. But cell types differentially express genes, well, differently, and traditional bulk RNA sequencing methods really just give you a singular data point per sample. With the scale and ease of use that 10X has enabled, You can profile tens of thousands of cells and multiple samples at a time and beautifully see these clusters and subclusters of cells within a mixture of cells in tissues or in fluids. And it's based all off of the individual cell's gene expression profiles. To the right is a two-dimensional view of a TCE plot where each dot is a cell, and it's clustered based on multidimensional information, and its similarity is clustered yet together. And the color codes are by known cell marker types. So just so you know what we're looking at here, we're going to show a few more graphs like this later on. We then took our single-cell expression functionality and applied that to the study of the adaptive immune system, where you can not only identify at the cellular level the subtypes of immune cells based on gene expression profiles, but also in T cells characterize paired alpha and beta chain receptor sequences, or in B cells, identified paired heavy light chain immunoglobulin sequences, giving VDJ recombination insights and clonotype information all in the same cell, which is critical for characterizing the adaptive immune system and helps us understand tissue microenvironments and how and what immune cells infiltrate. And we can't forget our favorite model system, the mouse, which we added to the portfolio earlier this year. Also earlier this year, we innovated on our single-cell RNA solutions and applied that to DNA. Our first application with this technology allows you to resolve copy number events at single-cell resolution, revealing CNV differences in heterogeneous samples, which are hard to resolve even with the most advanced informatics and bulk CNV sequencing analysis or in karyotype arrays. In this picture, a breast tumor sample was dissected and run through the single-cell CNV solution. Every line in the heat map represents a cell, and the coloring means a deviation from normal diploid cells. You can see there is variation with each portion of the tumor and within it. Further, on the top sample, you can see that there's a really small portion of cells that have copy number variation. And in bulk analysis, you may not be able to pick up those CNV events in that type of sample. And although this was applied in this experiment to tumor samples or cancer samples, it's also a tool that can be used to look at mosaicism in single cells. The 10x solution is now widely adopted, and ever since the launch of our first products in 2015, we've seen exponential growth in the number of publications, data sets, and software tools for a variety of application areas and research areas, which cover genetic health, oncology, immunology, stem cell, developmental biology, and population genomics. A set of publications that I'd like to tease out of that chart are focused on the understanding of cystic fibrosis, which notably is one of the most well-studied common diseases. One of the struggles with this disease is that although the genomics of the disease are well characterized, as are the end phenotypes, many patients with the same causal variants often have very different phenotypic outcomes in the main organs that are impacted by cystic fibrosis. In these two papers, single-cell gene expression was used to help identify disease-relevant cell populations to more directly link the resulting phenotype. The scale and cell throughput of using single-cell gene expression allowed researchers to identify a very small subpopulation of ionocytes in lung where CFTR is actually expressed. This deeper resolution and understanding can now help further pave the way for new avenues of biological studies. As the 10x portfolio continues to grow, we aim to better link genotype and end phenotype through intermediate and simultaneous measurements of phenotype at the ultimate resolution, the cell. And our new product developments, which we announced yesterday, truly support this. Now available for ordering include the single-cell attack solution for analysis of open chromatin and epigenetics. Also available for order is the implementation of the feature barcode technology, which allows simultaneous measurement alongside the single-cell gene expression solution and single-cell immune profiling solutions. And our biggest announcement is the availability of a substantially improved single-cell gene expression solution, a further improvement on our already industry-leading product. The single-cell attack solution allows for interrogation of open chromatin sites in single nuclei for a more resolved understanding of gene regulatory mechanisms. This ready-to-use solution performs just as robustly as any of our other solutions. This is a 3D TCE representation of cells and is clustered based on a new dimension of information like open chromatin profiles. Almost 10,000 PBMCs were analyzed, and the clustering of the cells labeled in the legend are very closely related to what you would find in single-cell gene expression. Here, we wanted to show the single-cell resolution of attack to a well-published bulk method. These are representative tracks of open chromatin regions from the GM12878 lymphoid blastoma cell line, and this portion of the genome represents a known region that has open chromatin sites. The upper orange track represents data gathered from about 50,000 nuclei in bulk, while the lower green track is an aggregate from over 600 individual nuclei run on the single-cell attack solution. Even with data from 75-fold fewer nuclei, the single-cell attack solution demonstrates very similar sensitivity to what you would see in the bulk approach. The lower box then shows the open chromatin sites, but at a single-cell level, from cell 1 down to 641. This is the same sample of what's aggregated in the green above. And as you would expect, each cell has a similar open chromatin profile as what you see in bulk because this sample has these known open chromatin sites. However, in more complex samples, we can resolve the different open chromatin profiles of certain cell types that bulk ATAC-seq just can't do. Back on October 3rd, we had a webinar with Dr. Howard Chang from Stanford University, who used single-cell attack on bone marrow samples. And he was able to resolve within cell populations different stage-specific enhancers. And he's really enthusiastic about the throughput and the scale of this approach. And if you are interested in viewing this, it can be found on the 10x Genomics website. The next capability that is now available for ordering is the enablement of the feature barcode technology with our immune profiling solution. The feature barcode capability allows you to simultaneously measure within a cell different feature information. The applications that we've started with are the capability to map TCR clonotypes to multiplex sets of peptide MHC molecules, which are antigen-presenting molecules, to understand T-cell specificity at a cellular level. The feature barcode technology also enables the simultaneous measurement of cell surface proteins for an even more resolved view of cell types and states. In this picture, you can see a specific T cell that has a specificity for a certain antigen and has this peptide MHC dextramer from Immudex bound to it. We then put those labeled cells through the immune profiling solution and are able to generate profiles that show the clonotypes specific to the antigens presented. In this case, cytomegalovirus and Epstein-Barr. And these profiles that you see here are very closely related or very closely matched to what you would actually see in flow. With the feature barcode capability, you can actually measure up to 4 different readouts per cell: gene expression, TCR or immunoglobulin paired receptor sequences, T cell specificity or B cell specificity to antigens, and cell surface proteins. A really huge step forward in true multi-omics approaches. In addition to having the feature barcoding technology as a part of our immune profiling solution, we also now have it available for our single-cell gene expression solution. The applications that can be enabled through this are simultaneous measurements of gene expression and cell surface proteins of the same cell, but also highly parallel functional genomics with simultaneous gene expression and CRISPR screening, or someone could do both protein and CRISPR experiments together. All of these applications are ready to use, but it doesn't mean that experimental designs are constrained in any way. And we really actually look forward to seeing what you can do with the feature barcoding technology. I do want to show you some data on gene expression and cell surface proteins. And here we have some data. It's 2 different TCE plots of about 10,000 human PBMCs. And they were labeled with specific antibody conjugates compatible with our feature barcoding technology. The antibodies were chosen in order to identify specific PBMC subpopulations. You can see on the left, PBMCs clustered in specific subclusters are expected, as you would want to see in PBMCs, based off of their gene expression information. However, see the circles up there? Okay, good. However, the double negative T cells in these circles here are not quite resolved in the gene expression TCE plot. And they're kind of mixed in with these memory T cells. When you look at the protein-based TCE on the right, the additional cell surface protein information allows for the double negative T cells to be clustered more closely together. Those are the blue dots. Another example that I don't have circled up here in these 2 plots are the additional resolution of memory and naive T cells with the inclusion of antibodies that were specific for CD45RA and CD45RO. And so now you can resolve those cell types with this combination of data, and it can also enable you to detect isoforms that are not resolved in just unbiased single-cell gene expression. The use of feature barcoding technology just really now adds this extra layer of dimensionality to these analyses. In high-throughput functional genomics, we show here how cells have lentiviral transduction of a multiplex pool of guide RNAs. The goal is to have 1 guide RNA within 1 cell. And then these cells go through the single-cell gene expression solution. And with the implementation of feature barcoding, we can assess which guide RNA, based on the protospacer sequence, and is in which cell, and then can look at the perturbations that happen in the gene expression profiles of each of those cells. And actually, Molly, one of our speakers, will be talking about this type of application today. The newest announcement, and the one that I am personally really excited about, is how we've pushed the boundaries of our single-cell gene expression solution with improvements in chemistry and consumables for up to 2-fold increase in gene sensitivity. We will also have new software releases, among other improvements, which has improved the detection of low RNA content in cells. In this set of data, our version 2 and version 3 of the gene expression solution was run with a complex cell type. You can see in the chart that the gene sensitivity is about 2-fold greater at the standard reads per cell. This improvement in gene sensitivity gives really greater experimental design flexibility. It can allow you to detect more genes per cell without the need for additional sequencing. Or you could increase the number of cells within the same experimental design costs. or achieve the same cell and gene metrics that you are used to today and have up to half the amount of sequencing reads needed per cell. Or you can kind of go the opposite way and sequence even more and continue to push the bounds of genes detected within your experiment. So these many dials are now available to you with this improvement in performance as you run the experiments the way you need to run them. 10x Genomics has really expanded the portfolio to add to its already well-adopted solutions. And we continue to push the boundaries of revealing the true complexities of biology by filling in the links between genotype and phenotype and combining modalities. All of these solutions are now available for order and consist of our Chromium controller, our reagents and consumables, and easy-to-use pipeline and visualization tools, making it straightforward for anyone to incorporate these methods into their experimental goals.